So I'd like to thank everybody for attending tonight to listen to Dr. Rose's presentation on the kind of latest and current research findings on the outcomes of the Arrowsmith program. So for me, it's really, really exciting. This has been a 40-year journey. Well, actually, it's probably been a 67-year journey because it's the journey of my learning difficulties and hunting for a solution and creating the solution and having hypotheses, and now the science is giving us some more information and insight into those hypotheses, answering some questions, and as research does, it's posing lots more questions that we are going to continue to investigate together and really try to understand more about this concept of neuroplasticity, cognitive stimulation, and how this can help individuals that are struggling with learning difficulties. So Dr. Rose is a professor with appointments in the departments of anatomy and physiology at Southern Illinois University School of Medicine, which is in Carbondale, Illinois, Southern Illinois. His research area is neuroscience, and he has long focused on understanding the mechanisms of synaptic plasticity, learning, and age-related memory impairments, which is really encouraging as I'm getting older and feeling a few memory impairments. He has over 125 refereed publications in scientific journals, as well as numerous invited book chapters. And I was just on ResearchGate this afternoon, and I don't know if this is accurate, but it said there's 9,036 citations in other publications of your work, and maybe it's even more than that. And over the years, he's received millions of dollars from national funding agencies to support his scientific studies. So really very grounded and solid in the whole research field. I met Dr. Rose several years ago. I think the first time we met was at the Neuroscience or Neuroplasticity and Education Conference in Vancouver that Howard Eaton and the Eaton Arrowsmith School group put on. The reason that we kind of intersected was Bremen Preparatory School in Carbondale took on the Arrowsmith program a number of years ago. It was Rich Collins who was the director at that time. He knew Greg. And when he took on Arrowsmith, he asked, would I be interested in doing some research? And I said, absolutely yes. So that started probably 5 or 6, maybe 6 years ago. 5 years. Yeah, 5 years ago. As I said, Dr. Rose is a consummate researcher. He's curious to explore questions in science to further our understanding of the human brain and its capacity for change. And tonight, I think the topic that he's going to speak on, research studies of the effects of Arrowsmith training on brain connectivity and neuropsychological measures. So without further ado, I want to hand it over to Dr. Greg Rose.
Well, thank you all for coming, especially at this late hour. The reason that this title is so long and somewhat vague is because we're really at the beginning of a very long road. So what I want to do today is kind of present some rationale and the results that we have so far that I think are actually kind of encouraging, certainly very interesting, but as is typical in science, have actually raised more questions than they have answers. So let's see if this part works. Okay, so, what are the goals of our work? Why do this research anyway? Barbara has empirically determined a program that really generates, I think, some pretty fantastic results across a species with also incredible variability, human beings. And, I think the proof of the pudding is how the subjects in Arrowsmith are able to improve their behavioral function in the dimensions that they want, depending on the type of exercises that she and her group determine are important for them. Well, basically my goal is a little more focused in that I am trying to apply, number one, more modern tools that let us actually look inside the brain and essentially predict its capacity to work in specific ways, and also try and build bridges to traditional neuropsychological assessments that are used in academics. These, of course, sometimes lead you down the rabbit hole. They're not necessarily important, but of course academicians think that they're tremendously important. So what I'd like to do is to be able to build a bridge between, you know, the empirical rubber meets the road, what are we doing and how is it working, and the kinds of mechanistic studies that neuropsychologists do, which hopefully can provide another dimension into the insights into the dimensions of how the Arrowsmith program works. So, you know, what we'd like to do is, you know, I want to understand from a neurobiological perspective how students benefit from Arrowsmith. And of course, the ultimate goal is, you know, you need to understand something if you want to improve it. I mean, you can't just kind of do things randomly. So, you know, we want to look at the brain mechanisms and you know, how Arrowsmith training influences these brain mechanisms. So potentially we have the ability to optimize the therapy or make it more efficient. I know I'm you know, on dangerous territory here because Barbara already knows she's got the optimal paradigm, but as a scientist, you know, one is always questioning. So I just listed here a few of the, you know, I think the premier questions to start with. And of course, the first is, using the tools that I have, can we see changes with Arrowsmith training? Okay, I mean, basically, an affirmative answer to these questions is very important because it tells us that our tools have sufficient sensitivity to be able to look at various manipulations and say, well, does one thing work better than another? And the tools that I'm using right now, I think, you know, the very most sort of front end one is looking at something called brain connectivity. And I'll be spending a few slides actually explaining what this is and how we've been able to use it using magnetic resonance imaging, okay, which is something maybe an MRI is something that maybe many of you have had already, but it tends to be something, a tool to take a picture of something. It might be your knee, it might be, you know, your shoulder, it might be your brain, but these are sort of static pictures. And the key word here is functional. Okay, this is a modification of the technique that really sort of gives you the possibility of watching the brain think, if you will, and I'll be explaining this. Secondarily, as I said, we want to build bridges to, you know, so the world of academics. So we're doing cognitive function assessments using what are our standardized neuropsychological testing vehicles. And these actually also kind of spill over into the area of education. In particular, what we've been focusing on are the battery that developed by the Woodcock-Johnson group. And, you know, again, many of you parents may be aware of these already because your children have been assessed using various components of this battery. The thing I am particularly interested in is building a bridge between any of these measures and the real world measures of success. And I think this is an evolving concept. We're really not there yet, in part because, you know, I'm here to talk to parents and students and figure out what it is, you know, how do people actually judge success? I mean, how do you think that, you know, why has Arrowsmith training been good for you? And, you know, this is something that initially I thought, oh, you know, we should just look at academic measures, standardized testing. And of course, it turns out that some countries have standardized testing and others don't. And every test is different. And so it's like virtually impossible to compare them. But it also turns out in my conversations with parents that I've seen that actually, you know, they're not necessarily putting the top priority on academic success. Instead, what they're interested in is sort of like how they themselves, if they happen to be participating in the study, or their children are kind of interacting with the world. You know, how well are they communicating? I mean, to put, you know, it in very, very the broadest terms, are they happy? You know, things like this. Again, you know, I'm not a psychiatrist, so I can't, you know, necessarily specify this dimension, but it's clearly something that's different and, you know, maybe parallel to academic success, but it shows that, you know, it's just performance on tests is not the only way to get along in the world. And in the long run, of course, it won't be. So, you know, I'm using these different goal measures to approach different goals. And, you know, theoretically, empirically, where we say, okay, changes in brain that we can measure, changes in brain circuits, provide the basis for changes in cognitive function, and these changes in cognitive function then will provide the foundation for improvements alterations in the real world measures of success. So, you know, I also am really sensitive to the notion that the kinds of alterations that happen in human beings as a function of training are very complicated, and they can take a long time. And especially from my experience with the students at Brem School, which tend to have complex multiple learning disabilities, you know, what one would like to know is, you know, spending time, 1, 2, 3 years in this program, going to be valuable? I mean, will there be results that anyone, either the subject or the parents or the real world, can be happy with? So can we develop an algorithm that will allow us to predict who will benefit and the degree that they will benefit from Arrowsmith training? And secondarily, right now, Arrowsmith has developed, I think, an incredibly sophisticated assessment battery. And so, you know, they also have sort of a programmed plan for how the students should be exposed to various exercises to correct what are our challenges in different areas. But frankly, you know, we don't have necessarily empirical evidence, or we haven't analyzed it, and I've been talking to a statistician here in Toronto who hopefully is making some inroads, or will make inroads on this. Is there a way that you can optimize the training, that you can speed it up? You can say, okay, listen, doing this particular exercise, if you do it 50 times, it's going to be just as good as if you do it 500 times, you know, where we can make the experience as effective and pleasant for the subjects as possible. So, the next few slides I'm going to talk about functional brain imaging. As I mentioned, the notion of brain imaging, it's taking a picture of the brain and you know, or some other body part. And this is really sort of an evolution of, you know, something that began decades earlier with X-rays, right? So with X-rays, you get pictures of bone, okay? But you don't see soft tissue very well. Well, because of the nature of the physics involved with magnetic resonance imaging, you get really great pictures of soft tissue. So that was the initial use, and it was great, you know, if you needed to look at the heart, or you needed to look at the lung, or you needed to look at the spleen, something like that. And you could actually get a picture of the brain that would discriminate between different brain regions and different types of tissue. Well, that was still a static measure. You could say, oh, well, you know, look, this person has had a stroke. I can tell where it is in the brain because I can see that there's a physical change, you know, a change in the coloration or a change in this picture. But it's not really telling, these static MR pictures couldn't tell you what's actually happening, how the pieces, different pieces of brain work together. So in functional imaging, what happens is that when neurons are active, so the neurons are you know, the functional units of the brain. When they're active, they use a lot of energy. Okay, so they use a lot of sugar, they use a lot of glucose, and there's a very interesting, not entirely understood relationship where neuronal activity then triggers the need for more oxygen, and that is it's signaled to, you know, to arteries, and so arteries actually then increase their output. They increase their delivery of oxygenated blood to the area where the thinking has happened, all right? And essentially, in functional imaging, what we do is we differentiate between blood that has lots of oxygen and blood that has less oxygen. And so the neuronal activity increases the blood flow, which increases the delivery of oxygenated blood, and that actually turns into the signal that we can detect that is like thinking. And so here, for example, a very small picture, you'll be seeing much larger ones that then, you know, so here's the brain seen from the side, and using false color, looking at the relative intensity of where the oxygenated hemoglobin is, then you turn it into a little color and you say, oh, well, that's the red spot. That's where in this particular task, you know, the activity in the brain is happening. So, you know, there are about, well, an infinite number of tasks that you can have a person do if they're in the MR scanner. And so that, of course, makes a research problem that's essentially insoluble. So, you can say these brain regions are important for doing this task, these other brain regions for that other task. Well, how can we get an assessment of what the brain actually has the capacity to do without having to test a million different tasks? Well, at first people didn't know. But it turns out that we know that brain regions have to work together to solve problems. I mean, as I'm sure you're all aware, well, you see something and it goes first to the place that that sees in your brain, but then actually it has to go other places where you say, well, gee, I'm seeing something. What am I going to do? What does it mean? What am I going to do about it, et cetera? So we know that brain regions have to work together. And it had originally been thought that this only happened when you were engaged in a specific task. Like right now, you're looking at the screen, and so you're saying, well, okay, so my visual cortex is registering this image, and I'm putting it together with what I'm hearing, and so this is forming a picture that turns into comprehension in some strange, unknown area of my brain. Well, it turns out that if you look at over relatively long time periods, not the sort of millisecond, fractions of a second interval that are used in specific processing, but over long times, okay, like in this case, 5 minutes, you can see that the signal that is the reflection of the brain oxygen slowly cycles. Very slowly cycles, very slowly, much more slowly than people would even have thought. And the interesting thing about this is it's cycling without you actually thinking, okay? That basically it's a resting connectivity. And so what people discovered, first this scientist named Biswal discovered that if you look at these cycles, which are happening all over the brain, that certain brain regions actually will cycle together. And so by looking at the different brain regions that were cycling together, they discovered what are called resting state networks. Okay, so you have networks because you have a bunch of different regions, and they're connected even at rest when you're not engaged in a specific task, and hence the name resting state. So here's an example of a resting state network. This is actually what it looks like if you're getting ready to, like tap your finger. So this is like the so-called sensory motor network, which includes sensory and motor cortex and something also along the midline. So this is a picture of the brain. It's like if you cut it this way and you're looking down from the top. Okay, and so the pictures I'll show you, I'll try and indicate what the plane is so you have a general sense of what's going on. But this was the first network that was ever described. And you might say, well, okay, fine, there are these networks and this is all well and good. What does that have to do with behavior? Well, what's really interesting, quite frankly, is that the strength of these networks at rest have been shown to predict how engaged the brain will be in a particular type of task when it's necessary. So the strength of the resting state networks actually predict task performance. And you might say, well, okay, fine, that's fine when it comes to like sensory motor function or seeing or hearing. But it turns out that there are actually a lot of different networks have been discovered. Right now there are 12 that most scientists agree on are separate networks. And there are those that are involved with sensory perception. But there are also these other networks that are really, I think, kind of interesting because they have more of a potential psychological import. So something that has to do with executive control, which is sort of a planning and execution function. Like you need to bake a cake. Well, okay, you need to turn on the oven, you need to, get out the cake pan, you need to get out the ingredients, mix them in a certain way. The salience network, which is something that actually sort of all the things you're perceiving in your environment now, you may have just heard somebody cough and you're saying, well, gee, that's not important to me. What's important to me is listening to Dr. Rosen, what he's showing up on the screen here. So it's a signal detection thing. There's an attention network, which is the part that really lets you perceive everything that's going on in your environment. So, you hear the roar out in the hall and you say, well, I wonder if that's a tiger. And it's unusual, so you say, well, yeah, in my salience networks, that's a tiger. And your executive function network says, uh-oh, I better run away. So this, I think, is very interesting in that it lets us get into more, the thinking part of the brain. So here we have, some cartoons of the brain from the side. Okay, so this is from the outside looking in, and this is from the midline looking out. And you can see that there are, hot colors and cool colors. And the point of this is to show you that actually that networks don't just act in isolation, that instead there are networks that actually, sort of like coordinate their activity together. So you can see, like the orange line and the yellow line, and you can see how well synchronized they are, again, over this very slow time course at resting state. But while these guys are, kind of like working together, you can see that these the network that's outlined here in blue is going in the opposite direction. So this is very important because it's telling us that actually there's an interaction between these networks, and that networks activating one network may turn off another network. And so that basically is very important in terms of regulating how the brain processes information, that it can do it in a sequential way. And I'm not saying that we actually have a complete understanding of how this process works, but we do know that there are networks that are correlated with each other and networks that are so-called anti-correlated with each other. Okay, one turns on, another turns off. And this seems to be just as important for normal brain function as having everything kind of like work together. So I told you that there were like 12 networks that have been, there's pretty much uniform agreement. Well, these are like the important networks right now. Trying to deal with 12 networks at a time is a difficult issue from a computational standpoint, or any, even, sort of the general thinking about how things work standpoint. So what we've chosen to focus on are 4 networks that have, I think, psychological import. And so this is how our analysis has proceeded so far. So these are the default mode network, the dorsal attention network, the salience network, and the frontoparietal control network, also known as the executive control network, or you may have heard of it as the cognitive control network. So I've listed them in this order, and you can see in each one of these cartoons, here's a picture of the brain, or a cartoon of the brain from the side, from the top, and from the front, and you can see the little colored circles here that identify the discrete regions in each of these networks. And you can see they're not just adjacent to each other, they're scattered all over the brain. And the reason that we're interested in these networks is because of, if you allow me a little bit of latitude here, we're looking at the sort of potential neuropsychological impact of what these networks do. So this default mode network is actually a very interesting network that people at first didn't believe existed, because it's most active when you're not doing anything. Like when you're sort of daydreaming, and the idea is that, you can sort of divide your universe into 2 halves. There's the half where you're actually attending to things, like you're hopefully doing right now, and then there's the half where you've gotten information and you're needing to process it. And so you kind of are how this is. You're like listening to your inner voice. You're thinking about stuff. So you're not paying attention. And this is where you get run over by the train, because you're not paying attention to the fact that you heard the signal of the whistle. But this seems to be really important for encoding memories. So then, as we talked about before, if we interact with the outside environment, I envision this as sort of a sequence. Well, first there's the idea of attention. Okay, what's going on in your outside world? Secondly, salience. Well, of all these things that I'm paying attention, or that I have registered in my brain, what's important? And then, of course, the idea is that you need to figure out what you want to respond to and how to respond. And that's the function of this executive control network. So in terms of our analysis, what we're interested in is both the strength of the connections within a network, okay, so like between the various nodes of each distinct network, but also between them, as I told you about the idea that the networks interact with each other. Sometimes one turns on, another one turns off. Other times, one turns on, another turns on with it. So what do the networks look like in a group of American high schoolers? Okay, well, so here is a group of, just over a dozen people, students, and you can see this actually shows the results of, well, it sets a template for an analysis I'm going to be showing you a lot more of, where we've done, a lot of computation to establish the relative connectivity, the strength of the connections between different nodes of networks. So I have the 4 different networks, and we have a cartoon, again, brain cut and looked down from the top, of where these different structures within a network are. So you can see that there are 4 elements in the default mode, 4 elements in the dorsal attention, 4 elements in the executive control, but the salience, which seems to be particularly important, actually has 7 distinct nodes that need to kind of work together. So in this analysis, then you can see this little colored bar, and the colored bar, the hotter it gets, the stronger the connection is at rest. The cooler it gets, the more blue, okay, the more unconnected things are. So you can see that in our what we call typically developing adolescence, that within each distinct network, that there's a lot of yellow, orange, red. All right, that these connections at rest are working together, and they're working together pretty strongly. So that kind of reinforces our idea that, well, yeah, these really are networks that hopefully have something to do with something important. But I think equally important, I'm going to now walk over here, and I know that photographers are going to hate this, but this is another way of looking at the network representation, okay? And it's kind of complicated, but I think it's informative that we just look at it from the standpoint of broad brush, okay? And the broad brush is, well, here are all these areas that comprise all the regions of the 4 networks we're looking at. And so here we have, typically developing controls, and you can see that there's a lot of red and a lot of, sort of yellow, where networks are positively connected to each other. But what's important, I think, about this is that you can see when we look at the interactions between networks, whereas within networks, everything is always positively connected to the other regions of the network, all right? But if we look across networks, you can see, hopefully up here, that there's a lot of blue, okay? So in addition to those within network connections that are strong, then there's also a strength in terms of actually being able to disconnect or disable networks, other networks, while one network is engaged. Okay, so that I think is a really important concept. So this is what we would use as kind of like a benchmark in a way. Okay, these are our typically developing controls. Well, what does it look like in Arrowsmith students? Okay, and here you can see, you can begin to appreciate why I use this image over here. Here on the left, what we have is an analysis that is it's a subtraction. It's sort of saying, well, okay, what actually is the difference between those typically developing controls that I showed you in the previous slide and the group. This was actually a group that was imaged, 16 students in 2017. And you might have thought, well, probably not that much is different. But look at this. I mean, it's just disgusting. It's so complicated. I mean, lots and lots of differences, different connections. And so that's why moving from, this top of the brain down, where the lines are so thick and there are so many, you can hardly understand them. It's a little easier to see things if you look over on this side, where again, things are sort of all spread out and it's easier to see the discrete lines. And so what you can see from this is that, somewhat surprisingly, there are a lot of red lines, okay? And the red lines indicate that the connections are actually stronger in the Arrowsmith students than they are in the typically developing controls. And you might have thought, well, gee, a brain that maybe isn't working as well, maybe it's not turned on as much. Okay, but it turns out that it's actually more turned on. And now, if you look over here, you can see that there are a few blue lines. Okay, and it turns out that all of those blue lines, those connections are restricted to the salience network. So we've got general overconnectivity, but within this one network, the sort of like, what's important network, that's not working as well, or it's not as strong as it should be. So, the net result was our discovery that, in particular, between the networks, there was what we're terming hyperconnectivity. Okay, that instead of the connections being weaker, they're actually stronger. And you can see this as I, look at the individual networks. You can see that overall, the within network connectivity is somewhat weaker. Okay, now the somewhat means that given the variability across students, when we analyze the entire population, that the differences, at least at this point with our sample size, didn't reach statistical significance. So, for example, like in the salience network, well, yeah, you can tell that there's a difference, that it actually is weaker, but it turns out not every connection is weaker. And so that's why it's not consistent enough to be statistically significant. We, in scientific studies, try and provide a measure of reliability using statistical tests. So here, what I've done is just to indicate with these little asterisks what is statistically different in our sample size. And you can see that actually, that of the 4 networks, 3 of them have statistically significant differences, but the difference is in the direction of overworking. Okay, not underworking. And even in the executive control network, you can see that there's a big difference, but it turned out that the variability is sufficient that with our sample size, it's not statistically significant so far. So this also, I think, is a fundamentally important concept, that it seems like the brain is trying to get the job done, and it kind of can't for some reason. And so it's just working really hard. It's like, you're spinning your tires, your car is stuck in the snow, so you're spinning, trying to get out, but you're just not going anywhere. And so this, to me, was actually the first big surprise, that the brains of Arrowsmith students are like working harder than regular brains. So now we can actually begin on this baseline to describe what happens as a consequence of Arrowsmith training. And those of you who know something about Arrowsmith know that within the full program, every student gets an individualized set of tasks, things to practice on. And this, of course, also is a scientific nightmare, because you like to simplify your life and have things all be the same, so then you can look at differences. So when Barbara suggested that we do some imaging of students in the cognitive intensive program, this seemed like a really ideal place to start from my perspective, because all of the subjects in the cognitive intensive program are just doing one thing. Okay, and this oops, sorry about that, let's get back. My description of the cognitive intensive program is approximate, as I was learning today. I mean, it's an unbelievably complicated task in a sense, but the bottom line is that you start in one place, you have to achieve mastery criteria, and if you achieve mastery criteria, you advance to the next level, so that it becomes progressively difficult, and it allows essentially infinite levels of practice and differentiation of the capacity of different subjects. So what happens with 6 weeks of sitting in front of the computer and rapidly typing numbers in at the bottom based on a clock face? Well, as you might imagine, the students overall get better. Well, so duh, that's like, I hope so. But what's more important actually is that if you look here on the right, you can see that the individual performance is quite varied. And this actually represents the data from 16 subjects, but you can see there are only six individual points because some of them represent more than one subject. They're overlapping, and I couldn't separate them. Well, so this is really interesting because, of course, what we're interested in, what's the difference between this guy who, like at the end of 6 weeks didn't improve at all, and this guy who started out at the same point and ended up with a score of 11. That gives us, again, a very useful tool to try and we would have a hypothesis and we would say, well, changes must be happening in the brain, and they must be happening differentially depending on the level of achievement of the individual student. So what do we find? Well, first of all, overall in the analysis, what you can see here on the left, again, this is our brain seen from the top, is you can see that there are a lot of changes that actually happen as the consequence of 6 weeks of training in the symbol relations task. Over here on the right, where you spread it out, what you find actually is that most of the changes that one sees are either within the salience network, the one that was deficient, or the connections between the salience network and the default mode network. So basically the what's important and the thinking network. So, we thought, okay, this is pretty neat. Number one, it's absolute evidence that yes, there are neuroplastic changes that are happening in brain, because now you can see concrete changes in these resting state networks as a function of this practice. The question is, well, what do these measures actually mean? Are they relevant to anything? So the first analysis we did, after seeing that there was some kind of effect, was to look at the difference between students that improved over time versus those that didn't. And here we just broke it out into groups that showed the least improvement. So, overall in this entire group, they only improved by the score of 2. So everybody started out at 2 and they made it on average to 4, versus the ones that went from 2 to 11. And, the hypothesis would be that there would be a different set of changes between the students who improved the least and the students that improved the most. Well, if you look at this brain image, okay, the computational image of the students that improved the least, what do you see? You see nothing. So these students did not show any changes, any significant changes in their connectivity, whereas the students that improved the most showed many changes. So again, even though this is not, proving the causality that one thing is directly related to another, it's sort of adding to the weight of the evidence to suggest that actually changes in brain connectivity could well be underlying the change in performance that you see, and that this would apply to individual subjects. So, to try and take this a step further and, try and build this bridge between what we're seeing in the brain and what that might actually mean, then actually Barbara had, I think, the brilliant insight here to use a set of standardized neuropsychological tests that academics might be able to appreciate. So it turns out that there are lots and lots of tests. I think this is one of the world's largest rackets, because you just see so many companies that have tests, and often they're overlapping, and and they don't really tell you what they're for, and they certainly don't tell you based on your score what you should do about it. But I'm kind of like new to this field, so it's a bit of an emperor's new clothes as I evaluate this piece of things. So what we did was to look, we assessed students, this was in the following year, in pieces of this enormous Woodcock-Johnson battery, so we wouldn't fatigue the kids to death. And so we looked at, first we need to actually look at how to control this pointer. We looked at things that have a little bit euphemistic names, like cognitive efficiency or processing speed, and then some subtests that are actually used to make these composite measures. So pair cancellation is a thing where you basically have to, you look at things and you have to cross out pairs. So you have to recognize, first of all, what the image represents, and then is it the same as an image you just saw? Things like this, and it's really a simple-minded test. I mean, essentially, okay, you've got 60 seconds. Cross out as many pairs as you can, and then how many mistakes did you make? How many did you cross out? How many mistakes did you make? So, I like the idea that if you use multiple assessments to create a composite, chances are better that you really have kind of tapped into what this core resource is. So if we look at these changes in the performance of these Woodcock-Johnson assessments, you can see that over on the subdimension that is supposed to be assessing cognitive function, that in fact there are several significant changes. In fact, in all the measures we looked at except one, which is sort of, I think it's supposed to represent a working memory measure, its numbers reversed. So it's kind of like if I would tell you, 157, and you would have to say back to me 751, right, where you have to actually manipulate the information. Anyway, this one didn't change, all these others did. But then we also had the idea, well, we should really try and hit a home run here and see if the symbol relations training would translate into something that might be a little closer, say, to schoolwork. And so Woodcock-Johnson has a part of their battery they call the achievement tests. Okay, and so achievement tests are supposed to theoretically bear some relationship to your ability to work in school. And you can see that after 6 weeks in this single task training, well, we've seen improvements in cognitive function, but we didn't see the overall, yes, there's a statistically significant improvement, but no significant improvements in individual tests. So, I mean, basically this would be the kind of thing you'd say, oh yeah, we saw a difference, but you'd say, is it a meaningful difference? And I would say no. So it's clear that, in this particular task for this amount of time, that again, we know we're modifying how the brain is working, because we see that in the functional imaging. And we seem, I mean, it's easy enough to kind of build a bridge and say, well, this is also telling us that means something in terms of how the person is behaving, how they're performing on a particular test. But, it's not like solving all the world's problems. What I did want to do with this was, again, try and sort of relate this to individual differences in performance. So here you can see with this second year, again, we separated into the least versus the most improved, and the least group was the same. Again, everybody started at a pretty baseline level of 2. This year, the group didn't have the number of outstanding performers we had in the first year, but still there's a difference between these. And if you look over on this side, you can see that if they're ranked according to who improved the least versus who improved the most, you can see that actually, interestingly enough, those who improved the least in the SIP measures actually got worse or didn't improve on these tests in the Woodcock-Johnson battery. Whereas the ones that improved the most, you can see, showed significant improvements in a couple of tests. They improved in all, but again, with our small sample size, I'm not willing to put asterisks on everything. But yeah, like in this particular measure of processing speed, you can see they just incredibly improved. Theoretically, the more asterisks you have, the better it is, unless you're a statistician, where you sort of say, well, hey, it just doesn't matter. But anyway, so this is another kind of reinforcing bit of information to say, well, yeah, how well you respond to SIP training will be reflected in the changes one sees in brain connectivity, and that also will be reflected in how your performance changes in these neuropsychological tests. So that was what we've done so far with the SIP program, and thanks to some of you who are actually here and or the parents, what we're doing this year is we're adding to our sample size, and we're actually simultaneously assessing both the brain connectivity issues and the neuropsychological measures. So we can, now we'll be able to make direct comparisons and also hopefully reinforce the conclusions that we've seen so far, which I think are relatively remarkable given the small sample size. So we, of course, would like to know, the Arrowsmith program is not, 6 weeks of symbol relations training. Instead, it's a very complicated program that can last years and years. So what we've begun is we've begun analyzing both using brain imaging and neuropsychological measures with students that have been in the academic program for 1 year or more. And where we stand right now is we have data only from the 1 year mark. And here we can see two populations. One that receives Arrowsmith training in Carbondale, and this is associated with conventional training that is provided by the Brehm School, their own little, unique mix of things. And then here is another group from Toronto. And the reason I bring these up is first to show you that, again, you look at these connectome rings and you can see they're not identical, and you might expect that since the programs are different at the two sites. But what's more important is that you can see that there's a lot of blue here. So, again, you might have thought, well, gee, everything should be getting stronger. But I told you earlier that, in fact, the problem with the Arrowsmith students is that the connections are like too strong to begin with. So what seems to be happening that's very interesting is that with the prolonged training, and again, the individualized programs, and this is a work in progress, but what you can see is that connections are toning down. Okay, so basically this hyperconnectivity that we see at baseline is reduced. And so it's kind of like, it's the most amazing situation. It's like the brain is working better because it's not working as hard. So that's kind of the idea. And so then if we extend this to the neuropsych testing, you can see that we really went crazy here, and we used a much broader battery. And so here are lots of cognitive tests. And what I wanted to point out here is these ones underlined in red, these are the tests that we used in the SIP program. And you can see, number one, that there are a lot of asterisks, so there are a lot of significant changes, overall significance, but also individual significance. What I thought was kind of interesting was that the pattern of changes for the ones in the SIP program, the tests we used, it's exactly the same. I mean, basically, so at the 6 weeks of symbol relations training produces significant changes in the exact same measures that persist and are seen after 1 year in the academic program. So that means a couple of things. Either it means that any of the exercises in Arrowsmith is sufficient to enhance the performance in these measures, or it means that basically the combination of measures is important, or it means that if you do symbol relations, well, that's an early and persisting change. Okay, and this I think is also a very important aspect of the Arrowsmith program that we need to reinforce from the standpoint of more rigid scientific criteria, the fact that not only do you see changes, but changes last. And so this is part of the the slogan, Arrowsmith changes your brain. And I have to say, we have some data that suggests that might be true. So that's one interesting aspect of the 1 year results with the neuropsych testing. The other interesting aspect is that if you look over here on the achievement side, whereas with the SIP program, we didn't see any significant changes. But in the 1 year program, you can see that there are a number of significant changes. So why is that? Well, we don't know precisely why yet, but is it a function of extended training? Is it a function of more complex training, training in multiple tasks? Is it a function of the brain, these changes in connectivity adjusting themselves to provide a better functional output? There are lots of options, but the thing I think that's encouraging here is that it says, okay, at 1 year, you actually are getting changes that theoretically would predict better performance in like academic and life environments. So, so this, I think, also very something that is encouraging. It's not the end of the story, and we can't say that, okay, we have proven this, but I think we've gone a long way down the road. So, just to summarize, the points that you, of course, probably mostly already know. First of all, that the Arrowsmith students are a very heterogeneous group, which, basically I think it's a credit to Barbara that she has been develop a program that identifies this heterogeneity and is able to actually address the specific issues of individual students. It also makes analysis a lot more difficult when you want to do, like scientific experiments. But on average, we know that Arrowsmith students perform less well than their typically developing counterparts in tests of cognition. So they're just not getting along in the world to the same degree. We know that their brain connectivity is altered. And in particular, what's noteworthy is this hyperconnectivity between networks, where the brain seems to be like trying to get the job done, but it's not. And so it's just working harder, like all the time. And that if we particularly focus on the individual networks we're studying, that the salience network in particular, the connectivity both between and within networks tends to be weaker. So, maybe if there's a pinch point in this whole process, it's that, the difference between, oh, I see all kinds of things, but I can't differentiate what's important, that is the blocking point that then prevents the brain from effectively processing the information, remembering things, deciding what to do. Right now this is pretty speculative, but it's based on the data that we have from our imaging work. We know that training in the SIP program actually produces pretty striking improvements, strengthens in brain connectivity, that this change is proportional to the improvement over the training interval, and again, the focus is on the salience network and the connections to the default mode network. So deciding what's important and sort of this rumination or incorporation process of storing what's important would be kind of like my over the thumb summary of this. That academic year participation in the Arrowsmith program actually reduces the hyperconnectivity that we see in students at baseline. So again, seemingly moving things in the right direction. And this is paralleled by performance in the Woodcock-Johnson test, and in particular with the one-year training, improvements in the tests that are supposed to be related tell us something about the capacity of a student to perform in an academic setting. So, as I say, this is just the beginning of a long journey, and but I think we've been able to make really some pretty interesting progress in just a few years. And, thanks to you guys, we'll be able to continue to make progress and hopefully, achieve some of the goals that I highlighted on my second slide.